All right, chapter 1, an invisible world. So before we begin, let's talk about the definition of microbiology. So biology, bio is life, and ology is the scientific study of something. So this is the science that studies living organisms. And micro essentially just means extremely small. Microbiology essentially studies microorganisms or microbes, which are extremely small living and it can even be non-living organisms that cannot be seen with the unaided eye. This term encompasses a wide diversity of things. If you think about it, microbes can be unicellular organisms or multicellular organisms. They can be prokaryotic or eukaryotic, which we'll talk for later. They can be heterotrophic or autotrophic, and that basically has to do with their types of metabolisms. They can be life-sustaining organisms. So these can be organisms that make different foods for us or keep our microbiome healthy. Or they can be life-threatening as well. So these might be organisms that cause diseases. Lastly, even our body is made up of microorganisms. So as we just alluded to, the microbiome is made up of microorganisms, and the cells that compose us, a multicellular being, are made up of cells, which are essentially little microorganisms themselves. It might not seem like it to us, but microbes span a wide range of sizes. If we look at this microbe scaling chart, we see a period character in Helvetica type 12-point size is about 500 micrometers in size. Let me get out my little laser pointer here. So a period at Helvetica type is about 500 micrometers, also known as microns in size. So the largest we're going to be dealing with is the amoebas or protists, and they can be up to 300 micrometers in size. So some of these can be barely visible to the naked eye. The naked eye just means you cannot see them. You can see them without the aid of a microscope. We have other protists as well. We have paramecia, we have diatoms, and please feel free to go ahead and take some time and read through the definitions of these different microbes. Euglena. Here we get to baker's yeast. Baker's yeast is about 10 micrometers in size. So baker's yeast is about 1/50th the size of a period. So these are orders of magnitude smaller than a period. And down here at the bottom, we start getting into bacteria. So Escherichia coli is basically the lab rat of bacterium. It's the most well-studied bacterium we have right now. It's found in the digestive tracts of humans, of animals, in the intestines. And it is roughly, it says here, about 2 micrometers. It's a pill-shaped bacterium, so it's about 2 micrometers by 1 micrometer. So 1/500th the size of a period. These baker's yeast and E. coli, we most certainly cannot see with the naked eye. So as you can imagine, almost all of these require a microscope to visualize as individual cells. Many are beyond the capability of the microscopes that we would use in class. So let's continue. Here we have other types of bacteria. Lactobacillus, we use those to make foods such as yogurts and other fermented products. Cyanobacteria are those groups of photosynthetic organisms that I study. We'll get into cyanobacteria later in this semester. Staphylococcus are bacteria that are found on your skin. Most are harmless, but some can cause disease. Now we're getting into viruses. Now, viruses are another level of size altogether. So here we're still, we're still hitting about the micron size with these bacteria. Viruses take a big leap down. Here they're about 0.3 micron. Here we see all the way down to 0.03 micrometers in size. So we went from 30 micrometers to 0.03 micrometers. So this rhinovirus is, what is it, 1/10,000th of the size of the protist that we were looking at earlier. So this already kind of gives you a glimpse as to how diverse microbiology is in its scope of study. So we're about to look at an extremely diverse group of organisms in this class. So much so that this class really only scratches the surface of what we know about microbes. In fact, a study done by Indiana University in 2016 predicted that there are 1 trillion species of microbes, 99.999% of which have yet to be discovered. So, of course, there must be a system for keeping track of all of these microbes and living organisms in general. And how we do that is we classify living organisms into different groups. Here you can see the organization goes kingdom, phylum, class, order, family, genus, species. For humans, that would be Animalia, Chordata, Mammalia, primates, Hominidae, Homo, and Sapiens. Each one of these groups, the members within that group become smaller and smaller. The genus species category is the full name given when referred to an organism. So we are Homo sapiens. So the naming of microbes has a certain convention, and this is very important for you to follow. So the entire genus species name is all italicized. The genus, the first letter in that name is capitalized. Everything else will be lowercase. Examples of this are Escherichia coli. Here we can see the whole name is italicized. The first letter of the genus is capitalized, but the C in the species name is not. Now, after the first time that you use the genus species in an article or a paper that you write or an answer that you give on an exam, you will then refer to it in this format. So the genus then becomes shorthand and it's abbreviated to just the first letter, still capitalized, period, space, then the species. And this throws a lot of people off because your word autocorrect will try to capitalize the C. Make sure that's always lowercase c. E. coli is named after the German bacteriologist Theodor Escherichia, and colon is where it resides, so that's where it gets its name from. Now here we have our second example, Saccharomyces cerevisiae. Saccharomyces cerevisiae comes from the root words myces, which is fungi, and saccharose, which is sugar, and cerevisia, which is beer. So this is sugar-eating fungus that makes beer. Saccharomyces cerevisiae, and as we can see, all italicized, capital S, lowercase everything else. Now that we've mentioned it once, we will abbreviate the genus to capital S period, cerevisiae. And finally, here we have Salmonella enterica, and it is named after Daniel Salmon, who discovered it, and entero is Latin for intestines. So here's your first checkpoint question. What is the correct way to write the following name? Synicocystis elongatus. And the spelling's all correct. We just need you to make the changes to the way it's supposed to be written down. And when you answer this question in the checkpoint questions on the assignments, make sure the convention is correct, whether it's italicized or not. I hope that was long enough for all of you to write down that question. Now let's get into the different types of microbes. Microbes are highly diverse. We have bacteria, we have archaea, fungi, protozoa, algae, viruses, and helminths. And we're going to try and briefly cover each one of these microbes today. Now, this is not an exhaustive list of microbes, but these are microbes that we will be covering in class. For checkpoint 2, I want you to name one reason why it is important for us to study different types of microbes. So try and come up with a unique reason why it would be important for us to study microbes. Feel free to look at some of those different microbes we had listed, or to wait until the end of this lecture and see the different type of microbes and what they do. Okay, so the first one is bacteria. So we're going to discuss traits that all bacteria have. First on this list is that they are unicellular. So bacteria can form complex communities with other bacterium, but they're still considered unicellular organisms. They don't require multiple cells in order to be, in order to function and be alive. They are also prokaryotic. Most of you should already know what prokaryotic means by now. You should have taken a high school biology course or some college courses. And if you don't recall, prokaryotic basically means this is pre-nucleus. A nucleus is a organelle or a membrane-limited area that within that membrane contains their DNA. Also, bacteria reproduce by something called binary fission. And this is an asexual reproduction, so it doesn't require a male or female or other sex type in order for this reproduction to occur. And for right now, we don't need to get into a whole lot of detail, but binary fission basically involves one mother cell. So imagine this one cell. And what it does is it forms a what they call an invagination, or these little dents about in the midpoint of the cell. And within those dents, you have a membrane that grows across. And so this, you see this gray area, this is a membrane. And so you get a membrane that goes all the way through. And these two compartments break away from each other, and two daughter cells form as a result of this. And we'll talk about this more during microbial growth, so don't feel overwhelmed about understanding that process. The other thing is they have circular chromosomes. Chromosomes are the main part of their DNA that exists in the cells, and these actually are a complete circle. So it doesn't have an end. One end just marries into the other. In eukaryotes, that's not the case. We have linear chromosomes where there's two ends to the chromosome. The other thing is their cell walls contain a compound called peptidoglycan. And their metabolisms are very complex, and they can function in a variety of ways. So they can be heterotrophic or autotrophic. And that means what they eat in order to get their carbon. So heterotrophic organisms, they have to eat something else that's already made organic molecules for them. So an example of heterotrophic would be humans. Autotrophic organisms can take CO2 from the air or other inorganic carbon and fix it, which means to create organic molecules from it. And the example that we're all familiar with autotrophic organisms are algae and plants. But many other organisms can do that. They also can swim with the use of a flagella. So if you look at this organism here, it kind of has this mouse tail. This is called the flagella in bacteria. So here's a review of what peptidoglycan is. And there's a lot going on here, so just bear with me. But if you look here, we have two different types of bacterium. We can have a gram-positive bacterium and a gram-negative bacterium. And these categorize a bacteria on the way that their peptidoglycan forms. So remember, this is that compound that's in their cell walls. And the book should have this capitalized. This is from OpenStax. Gram is named after the scientist who studied peptidoglycan. So this should be a capital G named after him. But as you can see, there are several compounds you need to be aware of. So if we look at this bottom caption here, we'll see that we have N-acetylglucosamine, and these are the little blue circles here, and it's abbreviated NAG. And then we have another compound called N-acetylmuramic acid. And these two are actually connected to each other through a pentapeptide and a tetrapeptide. So these little balls here, these little green ones and these little yellow balls are actually 5 amino acids in pentapeptide and 4 amino acids in tetrapeptide. If you do not recall what amino acids are, please review the basic 4 biomolecules of life. So again, we have these N-acetylglucosamines and N-acetylmuramic acids, and these are derivatives of carbohydrates or sugars. And the sugar would be glucose here, and again, the N-acetylmuramic acid is a derivative of glucose. The gram-negative peptidoglycan layer does not have this pentapeptide here, but rather these tetrapeptides are directly linked to each other. So gram-positives usually have a much thicker peptidoglycan with many layers, and whereas the gram-negative is a very thin layer of peptidoglycan. While we're talking about bacteria and their cell walls, let's discuss a little bit about their shapes. Shape is very important because it's often the first step in identifying an unknown pathogenic bacterium or any bacterium. There are 3 main different shapes in bacteria. There is coccus, which is Greek for spherical, so they can be spherical or ovoid, like in this first example here. And this first example, if you look carefully, you can see that little invagination of the cell wall here and here on many of these organisms, many of these bacterium, and that's the process of cell division occurring. So if you look at this organism right here, it's about to divide, and soon it will look more like these two right here. And eventually, these will break away completely to start a new cell again and start dividing again. So a coccus cell might often come in pairs, or it might even come in several chains linked together, and we'll discuss what that phenotype is in a moment or later. Number 2 here is bacillus. And bacillus means they kind of look bacteria-like. They're sort of pill-shaped or rod-like. And this would be the second image here. So this is an example of a bacillus bacteria. And this is a little confusing because some bacteria are actually also named after their phenotype. So we can have a Streptococcus pneumoniae or Bacillus cereus, and they get their names from their phenotypes. The next is spiral. And so a spiral is essentially a spiral. They have a corkscrew shape, and here we see in this last image a corkscrew shape. Okay, so for now, that's enough about bacteria. Let's discuss archaea. Archaea are unicellular as well. They are also prokaryotic, so they lack a true nucleus. They have more like a nucleoid region, so they don't have a membrane-bound chromosome. They also reproduce by binary fission, and we got to see some great examples of binary fission in that previous slide, which is an asexual form of reproduction. A meaning without in this sense of the word, the prefix. They also have circular chromosomes. So they're looking very similar to us as bacteria, but they're not bacteria. They're very different in other ways we'll get to later in this, this course. Their cell walls, for example, do not contain peptidoglycan. And these little dudes often live in extreme environments. So they're often categorized as what we say are extremophiles. Phile, it means lover of, so they're lovers of the extreme. And also, as stated in the textbook, they are not known to cause diseases. There are three main groups of archaea. There are the methanogens, so gen to generate, so this means that they generate methane. So during their carbon metabolism, they will end up, so while they're consuming compounds that contain carbon, their end product, their waste product, is something like methane. We have the extreme halophiles. So halo means salt. So we have salt loving, and they live in extremely salty places. And next we have the extreme thermophiles. And in this case, thermo does not mean cold, it means hot. These are hot loving, and they live in hot, often sulfurous water. Now, I have this article here because there are often many exceptions in biology, and this is one of them. So if you remember earlier, I said that from the textbook and many other textbooks and sources, they say that archaea are not known to cause diseases. And we're starting to punch some holes through that hypothesis. So this paper is a review article about how methanogenic archaea that live in our intestinal tracts, the methane itself might be a gastrotransmitter, as we see down here. And it says archaea are the only confirmed biological sources of methane in nature, and Methanobrevibacter smithii is the predominant methanogen in the human intestine. And this is linked and has a strong association between delayed intestinal transit and the production of methane, or in other words, constipation. So stay tuned on that. Okay, now let's jump to fungi. We're getting a little bit weirder here now. Fungi can be both unicellular or multicellular. So obviously, not all fungi is microscopic. They are what we call eukaryotic, so they contain a nucleus. So they have that membrane, and inside of that membrane is a sac that holds their chromosomes. They can do both. They can reproduce asexually or sexually. Their cell walls do not contain peptidoglycan. However, they do contain chitin. Chitin is the same substance that forms the exoskeletons of many insects. They are heterotrophic. So if you remember, hetero means that they're getting their organic compounds from something else, something different. So they don't produce it themselves like in photosynthesis. So even though you might see them as green, don't be fooled. They're not photosynthesizing here. Examples of molds include, or fungi include molds and also yeast. So remember the Saccharomyces cerevisiae? That was the fungi that eats sugar and makes beer. That is a fungi. So the yeasts that are used to brew beer and wines, etc., are yeasts. Okay, now let's discuss protozoa. So we're getting a little bit bigger here. Protozoa, remember, they were as big as, I believe it was 30 microns in size. Protozoa are unicellular or single-celled. They are eukaryotic. They can reproduce sexually or asexually. They can be free living, so that is they don't live inside of another organism, or they can be parasitic. The Plasmodium falciparum that you see in the upper right is a protozoa that actually causes malaria. Amoebas are mostly free living. They can be heterotrophic or autotrophic. Next is algae. Algae are typically thought of as being unicellular, although they can be multicellular as well. I'm not going to have that on an exam just because the textbook says one thing and I don't want to be contradictory there. So not all algae are technically microbial. And that's obvious for anyone that's gone and ate sushi before, because what do they roll up your sushi with? Well, they use kelp. Kelp is a type of algae, and it's multicellular. They can be eukaryotic, or they are always eukaryotic, pardon me. So they have a true nucleus. They can reproduce sexually or asexually. The cell wall is not composed of peptidoglycan. It's not composed of chitin, but it is composed of cellulose, which is a carbohydrate that is organized in a special way. They are autotrophic, so they photosynthesize and they capture their own CO2, or fixing. They fix that CO2 into organic compounds, such as cellulose and sugars and other compounds. So they're capable of photosynthesis. Okay, algae can be used for a variety of products. Again, they can be unicellular, technically, or multicellular. Cyanobacteria are sometimes grouped as being algae, but I wanted to point out that cyanobacteria are not eukaryotic, they're prokaryotes. So don't let the textbook confuse you on this one. They just happen to be green species that float in the water. And so a lot of people call them algae, but that's not technically true. If you look here at the bottles, you'll see that drink, and it has spirulina in it, which is a algae that has a corkscrew type shape. and it is edible and healthy for you, apparently. All right, now we're getting really small. We're going on the other end of the size. We're going to look at viruses. Viruses are extremely small even for a microbe. They are acellular, and we're going to talk about this more later, but they actually are not made up of a cell at all. Although if you look at this image of HPV, human papillomavirus, you know, if you're not very well aware of what makes up a cell, this is not a cell. And we'll talk about that later in the semester when we get to acellular pathogens. They are often pathogenic. Although there are so many viruses, not all of them are pathogenic to humans. But they require other life forms in order to fulfill some of their basic functions. So because of this, they are not really alive. They cannot reproduce or metabolize independently. Rather, they serve more as a source of genetic code that hops from one living organism to the next with some sort of a shuttle, or in order to you protect that genetic information until it finds a new host that can metabolize and reproduce for it. They are composed of genetic material, DNA or RNA, and they are surrounded by a protein capsid. We will never see viruses using typical light microscopes. The image you see here on the right is done with what's called an electron microscope, a very expensive piece of machinery. Next up are helminths, and helminths are a whole different horse amongst themselves. So we're getting a lot more complex with these helminths as far as cellular division of labor. Helminths are animals, and as such, they are multicellular, and they have nervous systems, and they are much more complicated than the other organisms that we spoke of so far, and that they have cells that have very specific tasks. They are parasitic worms, and they can exist either as roundworms or flatworms. Again, they're multicellular animals. They are only microscopic during certain life stages. And they are quite horrific, some of them. So if we look at this hookworm over here on the side, they can enter the host by burrowing through the skin. And these hookworms that are present in the southeast can be transmitted by people walking barefoot. Their microscopic stages might include their eggs and their larvae. All right, checkpoint 3. B. anthracis, Bacillus anthracis, is the bacterium known to produce the toxin anthrax. What shape does B. anthracis exhibit? Go ahead and pause the video again if you need to write down that question. We're going to leap forward here. All right, checkpoint 4. Here we have this article, underground lake of liquid water detected on Mars. Recent satellite data indicates that there is a large amount of extremely salty liquid water underground on Mars. If this water were inhabited by a microbe from Earth, what type of microbe would it likely be? So think of the different microbes that we listed in this discussion, and you tell me what type of microbe it would most likely be. All right. Now the final checkpoint, checkpoint 5. An unidentified microbe is found to reproduce asexually, contain a nucleus, and possess a cell wall made of chitin. What type of microbe is this? Okay, so let's talk about why one would want to study microbiology. So one important role that microbes play is in a process called element recycling. Microbes are the only type of living organism that can capture atmospheric nitrogen and convert it to a form that is usable for others. Nitrogen makes up about 70% of the gases in our atmosphere. This is the case because it is an extremely stable compound that is very hard to oxidize. So it comes in the form of N2. However, nitrogen is one of the four main ingredients that comprise biological organisms. We need it because there's an essential biomolecule called amino acids that make up proteins. And so living things need this nitrogen in a usable form in order to survive. And there are certain microbial species that are the only living organisms that can convert atmospheric nitrogen, or N2, into usable oxidized forms in amino acids. So if you think about humans, we can't breathe in nitrogen and make amino acids. We're not breatharians. We consume products that have proteins in them, and we break those down into their constituent amino acids, and we use those amino acids to build as building blocks. This is also the reason why you often use nitrogen fertilizers, because the nitrogen fertilizers contain some sort of an oxidized form, or some sort of form where the nitrogen is fixed from its atmospheric N2 form, because it's really hard to get enough nitrogen in the soil. Another thing we use microbes for is sewage treatment and bioremediation. And this is where microbes can break down contaminants and toxins found in sewage or waste from industrial processes in order to make them more environmentally friendly. Next up are food and drink. Microbes are essential for producing a lot of our fermented foods and beverages. This includes yogurt, pickles, soccer nut, beer, wine, chocolate. The list goes on and on. And they are very important in newer forms of biotechnology besides food biotechnology. And these are processes such as genetic engineering, medical usages such as producing human insulin. We produce human insulin typically in yeast or animal cells in cell culture. Industrial products such as biofuels, plastics, food additives, and material biosciences, and for providing gene therapies for humans. Here we have Luxturna. This is a treatment for an inherited retinal disease that causes seeing issues in humans. And this gene therapy actually uses what's called an adeno-associated virus. And this is a type of virus that is benign to humans, but we use this as that shuttle. Remember, we talked about viruses needing a shuttle, and that's usually what you see them as when you see them in pictures, is they use this adeno-associated virus as a shuttle to move in genes that correct that mutation in humans with this disease. Microbes are very important to us in our human microbiome. So the human body is composed of about 30 trillion cells, and it is believed that they cohabitate with about a 1-to-1 ratio. So there are about as many microbes as there are human cells. So perhaps the most intimate way that microbes affect human welfare is through the human microbiome. Imagine if you took away all of the human cells of the human body and leaving just behind just the microbes, you would still be able to find your way around that body. Microbes are essential for digesting our foods. They provide us with vitamins. They prevent other pathogenic microbes or invading microbes, the bad ones, from taking up residence. They also are believed to be involved in certain signals to our brain and to the rest of our body. The microbes that live stably in and on the human body are called the human microbiome. So yes, like I was saying, the microbiome is seeded at birth and maintains our health throughout life. Here's an interesting study on that. The title is Partial Restoration of the Microbiota of Cesarean-Born Infants via Vaginal Microbial Transfer. And they show that babies that are born of a C-section are underrepresented in certain microbial population. Okay, so let's discuss Synlogic. Synlogic is a biotechnology company, and what they're interested in doing is taking bacteria and giving them the ability to produce certain compounds, whether that's enzymes or other compounds that that human bodies might need. And you see in the in the left column, there are a series of different drugs in the pipeline that help correct metabolic diseases that are seen in humans. And in the different columns, you can see we have screening, lead discovery, lead optimization, IND enabling studies, and now some of these are actually entering into phase 1, into phase 2 trials to see if we can use these microbes in order to recorrect these inherited genetic disorders, or possibly even cancers. So an example of this would be the urea cycle disorder, which is characterized by excessive ammonia production in the blood and a genetic inability to break down that ammonia into urea. So, very interesting. This was an interesting paper that came out about building the bio-based economy. So in biotechnology, we use feedstocks. So this could be biomass, which could be forest byproducts from the logging industry. It could be algae, methane, waste, and off gases from plants. And we feed those into industrial biotech cultures. So we grow big vats of culture. So instead of making beer, you're growing microbes to make something else. So this involves some sort of synthetic biology where these microbes are engineered with different enzymes, and then you go through a biorefining process. You might need further gene editing of these microbes, but ultimately we're going to build products. So there are many different products we can make. We can make sustainable fuels. We can make industrially relevant chemicals. We can make different polymers such as plastics and other coatings and things that are used in industry, and also food ingredients. So just as we can make foods, we can just as well as make ingredients for other foods. And this is very big business. I think a lot of people would be surprised by these numbers. So here we're going to look at revenues for the US company, for the United States in 2012. And if you look at this pie chart, it's broken down into 3 categories. We have red biotech, and these are called biologics. And these are complex organic molecules made from living organisms, from microorganisms, such as the insulin we discussed, or it could be some antibodies that are produced, or a whole slew of different things like the adenovirus gene therapy we discussed. That has a $91 billion of revenue in the United States. GM crops are not really microbial in nature. They can be, but for the most part, this is like your GMO corn and soy, etc. Next, we have industrial biotechnology. I think this might be surprising for some who think that medicine is kind of the blockbuster for revenues, but actually industrial microbiology brings in over $105 billion as of 2012. It is also the fastest growing industry amongst the three. So that wraps up today's lecture. We will finish up chapter 1 in the next lecture. Thank you all for being patient and listening to the video, and I hope to see you in the next round. If you have any questions, feel free to email me, and also remember to submit your answers through the quizzes portal on Canvas. So this would be checkpoint questions 1. Thank you.